Hello students, welcome back to Unacademy classes. I am Arunar Nair, your botany faculty. Today I come with another topic, that is cell cycle. Very interesting topic, and in your NEET examination there is a huge weightage also there. I believe that almost you can say that from this chapter, cell cycle and cell division, they always ask 3 to 4 questions. Got it? So very important topic. So today we'll do a quick revision of this topic, that is cell cycle. So what exactly is cell cycle? We know that every living organism have a natural birth to their natural death. Birth to their death. Okay, natural one. So it's a process where they will live. They do the metabolism, they do the growth, they do the development. Got it? Now here also, when we go to a single cell, because every living organism is made up of what? At least a single cell. Okay, and that when we talk about a single cell, the cell also have a cycle, where it will do what? It will have a birth, then it will grow, and after certain growth they will again divide. Do all cells do the same process? They follow the same pattern that they show the growth and then divide? Not necessarily. Some cells at specific location they perform this type of function. At certain cell you can see that they stop after that dividing. Got it? Here we will talk about the complete cell cycle, how it exactly works, and how in your NEET examination the questions will be asked. Got it? We'll start with the first thing, what you may see in the cell cycle. So in cell cycle we can divide this into two, interphase and M phase. You can clearly see this one, this entire thing up to here, from here to completely here, it is what? It is your interphase. So you can see that it's one of the longest phase in a cell cycle. So nearly, if I tell you that if it is like 24 hours to take a typical erythrocyte to grow in a lab, then almost 95% of the time it will remain in which phase? In the interphase phase. Got it? And only for few time only it will be in a divisional phase, that is the M phase. The M phase, what we have shown here particularly, is a mitosis. You can see the stages of mitosis, that is prophase, metaphase, anaphase, telophase, cytokinesis. So we have only mitosis as a cell division? No. We have mitosis also and meiosis also. Mitosis is an equational division. Whenever the cell has to divide, and it has to divide equally, it has to distribute genetic material equally, that is the equational division, that is your mitosis. But whenever you want to do the reduction in the genetic material, like from diploid to you want to make it haploid, at that time it will be a meiosis. So here particularly we are looking at mitotic division. So without further wasting, we will enter into the phases, that is the interphase. Now when we look the interphase, entire interphase again we can divide into their sub division, that is G1 phase, S phase, G2 phase. G1 phase is also known as gap phase. So it's a gap between the division and the synthetic phase, that is the S phase, which is very important. Why S phase is very important? Because this is the stage where the DNA replication takes place, where the genetic material will get doubled. So before that there is a gap phase, and this is the G1 phase, which is also can be called as what? Post mitotic phase or pre-synthetic phase, like that different names are there. But this G1 phase, what important thing we can see? Whenever newly formed cell is there, that newly formed cell will initially show a growth. You can see there is a plasmatic growth is there. So here the cell will grow. So initially when the newly cell are formed, it will have a very minimum number of organelles. So now what it has to do? What it has to increase its number of organelles. Then you will see the enzymes and proteins, which is essential for the metabolism, that all will be synthesized in the G1 phase. Also please keep it in mind that next phase is what? That is your S phase. So in the S phase, what's the important event is going to take place? That is DNA replication. So in your DNA replication, some of the important enzymes are there, like DNA polymerase, RNA polymerase. All those essential enzymes will be synthesized where? Obviously it will be synthesized in the G1 phase. So just keep it in mind, all the proteins and enzymes which is required for the metabolism will be synthesized where? It will be synthesized in the G1 phase. Number of organelles will increase. So cytoplasm is growing. So you can clearly see that the cell continues to grow, and that's what we see in the G1 phase. Next phase, that is the S phase. That means synthetic phase. What is the importance? Very important thing, that is DNA replication. If cell has to go for cell division, so definitely it has to have a extra amount of genetic material. With one marker I cannot give you two markers. First of all I have to make it what? Two marker. Then only I can distribute it to equally. So similarly here also The genetic material which you see, that is initially it will be like 2n, or we can say the amount of DNA is 2c. So if you want to distribute equally into 2 cells, so you have to have another set of what? Chromosome. Another, we can say, amount of DNA has to be double. Where this will take place? It will take place in this particular phase, that is the S phase, that is DNA replication. So what happened in this DNA replication is what? The amount of DNA, which is 2c, it will change to 4c. So the genetic material which you see, that will get what? Doubled. So just an example, suppose 20 picogram is the DNA amount, that 20 picogram amount will get what? Changed to 40 picogram. But what about the chromosome? Do the number of chromosome also increase in the S phase? No. Please keep it in mind, question will be asked. Like where the amount of DNA will get double? The answer is what? S phase. So what about the number of chromosome? Do it get doubled? No. Just imagine for a while. If chromosome looks like this, fine. So these are the chromatid. After condensation, only you will see at the time of your cell division. So just imagine for a while if chromosome is this. So right now it will be what? 2n. Okay. The amount of DNA is what? 2c. What will happen after DNA replication? After DNA replication, the same chromosome which you see, it will develop a sister chromatid. So what will happen to the amount of your DNA? The amount of DNA will become 4c. But the number of chromosomes is still the same. So that's why it will still be 2n. So can I see chromosome in S phase? No. Please keep it in mind. It's an interphase. During the interphase, the cell will have nucleus, and inside the nucleus, all the genetic material will remain in a chromatin fiber form. Okay, in a tangled form that will be remain. So I can't see chromosome in the S phase. Got it? So now we understood about how these things will work in S phase. So in S phase, DNA replication takes place, amount of DNA will get doubled, but not the number of chromosomes. So where exactly the number of chromosome will increase? That's a question mark. We know the answer, that is Anaphase. So in Anaphase, the number of chromosome will get doubled. I will explain. Quickly we'll look into that also. So what else can take place? That is histone protein synthesis can take place. Centriole duplication takes place. In case of animals, we know that there is an organelle called centrioles. So that centriole duplication will also take place where? In the S phase. So these all are the very, very important event that we need to keep it in mind, because in exam they will ask question. Next is another gap phase. So this is your post-synthetic phase or your pre-mitotic phase, that is the gap phase, G2 phase. Very important sequence, please keep it in mind, G1 as G2. Always you have to follow the sequence, very, very important. Don't juggle it because marks will be gone. Then what will happen in G2? Since cell is growing, so it will continues to grow. So cell will continue to grow here. So more plasmatic growth, or we can say the cell will achieve maximum cytoplasmic increase in this particular stage. And it will synthesize all the necessary proteins which is required for the cell division. So rest of the proteins which is important for the metabolism, for the cell division, all will be synthesized here. For example, one protein we can take example, that is tubulin protein. Tubulin protein will be synthesized where? In the G2 phase. Now tubulin protein, you know, the spindle fibers. Spindle fibers are made up of what? Tubulin proteins only. And spindle fibers, what's the function of spindle fibers? The spindle fibers do what? They arises from the centrioles and come to where? Come to the kinetochore of the chromosome, so that it can get attached and it can segregate the chromatid, sister chromatid or centromere. Very important, so that it can do the equational division. So that important protein will be synthesized where? In the G2 phase. So all these things, like in G1, cell growth, number of organelle will increase, synthesis of enzymes and proteins. In S phase, DNA replication, histone protein synthesis, centriole duplication. G2 phase, cell grows to continue, okay, continues to grow, and protein synthesis, tubulin protein, that very example we can take, that formation all will take place in this respective stage. Once you will see, will reach to a certain stage where it will achieve its maximum growth. So its nucleus and cytoplasmic ratio will get disturbed. Nucleocytoplasmic ratio is there. That ratio will get disturbed. Then it will give a signal that, okay, now you have to divide. Now cell will go for the divisional phase. That is the M phase. Here particularly we are looking for mitosis, equational division. So in the mitosis, the M phase, we can say it is of two types. Karyokinesis and you can see cytokinesis. Karyokinesis means nuclear division. So in this nuclear division, we again have a substages like prophase, metaphase, anaphase, telophase, and all these stages you have to follow the order. Very, very important. Then finally we'll come to the cytokinesis, that is cytoplasmic division. In cytokinesis, what we have? Prophase. So what will happen in the prophase? Nucleus you can see. So some of the events like nuclear membrane disappearance, nucleolus disappearance, endoplasmic reticulum, Golgi apparatus, all such organelles will start to disappear. At the same time, the genetic material we say that which will remain in a chromatin fiber form, that will start to what? Condense and form what? Chromosome. So you will slowly start to see the structure of chromosome. So condensation of chromatin material, that will all start in the prophase. So by the early prophase, all these events will start, and by the late prophase, you will see it will all get end. That means all the nuclear membrane will disappear, and endoplasmic reticulum apparatus will get disappear, and the central who started to move towards the opposite pole, it will almost reach to the opposite poles, where at the end of the prophase. Then next stage is what? That is your metaphase. In metaphase, what will happen? In metaphase, you will see all the chromosomes will get arranged at the center of the cell, and that center we call as metaphase plate. Question can be asked, and respective spindle fibers from the opposite pole will get attached to the chromosomes. So if I show you, suppose this is your chromosome, and here you have the kinetochore. So the spindle fibers will come from the one pole, and another from here it will come. So you see the kinetochore of both the end of the sister chromatids will get attached to the opposite poles, and this will take place where? In the metaphase. So in metaphase you can clearly see that the spindle fibers come and get attached to your kinetochore, and in this way this stage will be over. Do the number of chromosome increase here? No, still the number of chromosome will remain same, that is 2n. So what will happen in the next stage? Anaphase, very important, segregations of centromere. Sister chromatid will get separate. So here if you see, these sister chromatid at centromere which you see, it will get segregated here in this way. So the segregation of centromere, segregation of sister chromatid takes place in the anaphase, and it will start to pull towards the opposite pole, and aggregation of all this chromosome will take place at the respective poles. Here you can see the one chromosome will become two chromosomes. So the number of chromosome will get doubled where? In the anaphase. So question will be asked here where the number of chromosome will get doubled. Numerical type of such questions, like suppose the 14 number of chromosomes are there. So when the number of chromosome will get doubled? So answer will be anaphase. So please keep it in mind. It's the anaphase, not the S phase. Common mistake we do that number of chromosomes also we get increase in the S phase, but no, there only the amount of DNA will get doubled. The number of chromosome will exactly get doubled where? In the anaphase. Followed by the next stage, that is the telophase, where the telophase you can see it's a reverse of prophase. So the appearance of nuclear membrane, because all the genetic material reach to the respective poles, so around them you see the nuclear membrane start to reappear, organelles start to reappear. So it's just a reverse of what? Your prophase. Followed by the last part, that is the cytokinesis, where the cell division will take place, and you can see the two cells has been formed. Again it can continue, again it can follow the same G1, S, G2, again division. So you will see, do all cells follow the same pattern, like every time division, then again G1, S, G2, then again division, G1. So continuously they do this? Not necessary. There are certain cells who are present at specific location, they have a function to do, they can say they continuously they divide. For example, in plants at the tip part, meristem region, they have meristematic cells. We have studied that the meristematic cells do what? They continuously divide. So there we can see this S phase. G1, S, G2, then division, G1, S, G2. So they continuously divide like this. But there are certain cells who will go for what? They become permanent cell. Now those who become permanent cell, they won't able to do the cell division because they lose the property of cell division, and they will enter where? They will skip the G1 and they enter to the G0 phase, which we say quiescent stage also. So in the quiescent stage, the cell will be metabolically active. Got it? The cell will be metabolically active, but no longer it will do the cell division. So that means it will be a living cell only. It will do all its metabolism. Its specific function is there. It will perform all those functions, but it won't able to do the cell division. Like for example, heart cells and all. But there are, on the other hand, there are certain cells who will go for G0, and at any point of time, based on the requirement, they can again come back to the G1 and it can follow again these all stages. Like for example, your hepatocytes, liver cells. Those liver cells are there. We can see that any damage happened to the liver cells. So what will happen? The nearby cells will get the signal, and all those cells will do what? They will replace all that damaged cells. And they are already in the G0. They will come to the G1 again as G2, because when they come to the G1, then the next phase is the S phase, DNA replication. Once the DNA replication happened, they have to divide because amount of genetic material got doubled. Then they will go for G2, division, this followed. Once the cells are replaced, then they will again go back to the G0. So in this way, every cell will do the cell cycle, and there are certain cells, like I said, they will follow the cell cycle every time. There are certain cells are there, once they divide it, they form and they will escape out from the cell cycle and they will remain in a suspended in the vehicle's G0 stage. Got it? So this is all about a quick recap of the cell cycle, where we discussed about the interphase, one of the longest phase, or it's a resting phase also, because when you look through microscope, you won't see anything, but metabolically very, very active stage. Then we have the M phase, where we have nuclear division, karyokinesis, which have substages like prophase, metaphase, anaphase, telophase. Then we have the cytokinesis, that is the cytoplasmic division. In your NEET examination, very, very important, all these things which are there, these events which are mentioned here, from here only the most of the time they ask the question. For further details, for more understanding of the topic, please join our Unacademy offline center Kochi. We are waiting for you. All the best, students.